Okay, so hi everyone. It's great to virtually see everyone. It's been a little while. Um, I I'm presenting two articles that kind of touch on the idea of predispositions to myocarditis. So I have no financial disclosures, and this presentation does not reflect the opinions of my employer. Here are the two articles that I'm going to present. The first one is authored by Amy Kontorovich, and it's myopathic cardiac genotypes increasing the risk for myocarditis. So I'm going to be going over that one first. The second one is pathogenic variants in cardiomyopathy disorder genes underlying pediatric myocarditis. Are there further impacts of a heterozygous immune disorder gene variant? And let's get started. So all of you know that acute myocarditis, it's long been considered more of a kind of a random process, kind of unfortunate, and it's a consequence of whether it be infectious, toxic, autoimmune reactions to the heart. Pathogenic organisms are usually implicated, so usually it's viral. We're talking about adenovirus, Coxsackie, parvovirus B19, and herpes viruses. And usually these cause no more than an uncomplicated respiratory tract infections in most people. However, as we all know, it can cause sudden cardiac death or permanent heart failure in a subgroup that develop acute myocarditis. The outcome from the viral infection is therefore thought to be somewhat of an outlier, given this kind of idea of randomness, that it's only affecting random people. And furthermore, this idea is suggesting that there are some sort of genetic factor of the host that might influence pathogenesis. So maybe it's not actually that random. So this notion of randomness and acute myocarditis susceptibility, it's further challenged by the following studies that are cited in this paper. There are more studies on this, not tons, but there are more studies, but these are the ones that they have discussed. So the first bullet point talks about some studies that talk about acute myocarditis being more common in individuals that have a genetic mediated derangements in the cardiomyocyte cytoskeletal components that include like dilated cardiomyopathies of Duchenne muscular dystrophy or an arrhythmogenic cardiomyopathy. In another study in children with acute myocarditis, there's an enrichment in one of these studies in rare biallelic non-synonymous or splice site variants among a set of genes to several cardiomyopathies, and you can see the genes listed in the parentheses. Now, this particular study was actually done by the same author that wrote this paper. And then lastly, likely pathogenic and pathogenic variants were actually found in a overlapping set of cardiomyopathy genes, and those are listed there in the parentheses on a targeted gene panel testing in 5 out of 8 children that were in the hospital for acute heart failure. And this was caused by acute myocarditis, and this is by Brown et al. So these discoveries kind of cast acute myocarditis as more of a human genetic disorder. Maybe there's some genetic overlap with the myopathic diseases of the heart. Now, I would be remiss if I didn't mention the final common pathway. So the final common pathway is highlighted in this paper. And about 20 years ago, when the gene discovery for Mendelian genetics was in its infancy, there was a paper by Bowles et al. And he proposed that heritable conditions with similar phenotypes and genetic heterogeneity, they actually occur because the abnormalities in genes that encode those proteins have similar functions and they're involved in the same pathway. So that it's kind of common sense, but this is what we call the final common pathway. Now, a few other studies kind of support this. So there's a study with genetic cardiomyopathies that were broadly linked to derangements in proteins with the sarcomeric and cytoskeletal structures. This actual hypothesis was repeatedly upheld in a discovery that implicates proteins in the sarcomeric structures with also nuclear membrane, Z discs, desmosome, spliceosome, and ion channels. And I just left all these numbers in here because I figure you guys can look. I mean, you can get in a really big rabbit hole. There's such great papers and sites. So definitely check out some of these references here. And then lastly, the expert consensus that classified that there's a core set of genes for cardiomyopathy. And then there's a similar phenotype of cardiac dysfunction that accompanies a skeletal myopathy in several genetic neuromuscular disorders. So the hypothesis that we're working with here is that the vulnerability to acute myocarditis is caused by a genetically mediated impairment in proteins and a range of cardiomyocyte cytoskeletal structures caused by variants in so-called cardiomyopathy genes, as well as the neuromuscular disorder genes that have a myocardial expression. So we don't want to forget those. So this is what they call their visual abstract. I'm not going to go into very detail on it, but I like to show this because now I'm going to go over the methods, and then I want you guys to appreciate kind of how they structured their cohorts. So up here on the left, you can see, let's see if I can get my laser pointer. Up here on the left, you can see that this represents the acute myocarditis cohorts, of which there are 3 separate cohorts, and I will go into that. And then they have, they very carefully chose controls for these studies. So they have these heart healthy controls that were matched by sex and ancestry. And then once they, got all of this together, then they did the testing. So they did targeted panel sequencing. Viral PCR was actually done as well. And then they ran all their statistics. So here are the methods. So for subjects with acute myocarditis, there were 3 different registries. The registries were labeled as AM1 for acute myocarditis, so AM1, AM2, and AM3. AM1 cases were actually adults and children, and those had a clinical diagnosis of acute myocarditis. This was based on both historical and diagnostic testing data that were recruited between the years of 91 and 2000. Then the 2 other cohorts, AM2 and 3, those were subjects that had a clinically suspected viral myocarditis, and those were recruited from 2015 and 2017, and then also 1997 and 2013 periods. And this was, myocarditis was actually proven by the immunohistologic criteria. And so here's a little snippet from that actual citation, from that reference 19. And the immunohistochemical detection of focal or diffuse mononuclear infiltrates with greater than 14 leukocytes per millimeter squared. And the leukocytes would include CD3 positive T lymphocytes and or CD68 positive macrophages. And, as we discussed in one of our meetings, sometimes CD3 will also stain macrophages every now and then. But in the myocardium, they were looking for this in addition to enhanced expression of HLA class 2 molecules. And so they had a actual immunohistochemical stain that stained for the HLA-DR alpha. So I've never actually used that, but that's what they used. Now, the DNAs that were provided for all the subjects from AM1 and 2 and samples from AM1, they underwent a whole genome amplification prior to their sequencing. And then for subjects from the AM3, genomic DNA was extracted from frozen banked whole blood specimens. And there's more kind of details on the specifics in the paper. Now, we kind of briefly touched on their control subjects, but these were identified through a previous enrollment in the ICANN School of Medicine's BioMe Biobank. And it's a special protocol that's approved by a particular IRB. And they go into how they accounted for the ethnic heterogeneity of the cohort. It says the first 3 principal components of the analysis from sequencing data on these cases and control studies were used to generate a subset of genetic ancestry and sex matched controls. And in these particular cases, they lacked relevant acute myocarditis, cardiomyopathy, heart failure, and neuromuscular disorder ICD-9 and 10 codes. So if these cases actually had a lack of those codes, they selected those as appropriate controls. And this was from the electronic medical record at a ratio of 4:1 to each case per cohort. Now, the genetic analysis, I won't go into the nitty gritty, but we have our next genetic sequencing that was performed on the genomic DNAs. And like I mentioned, it was a targeted panel, so of 93 genes. And I'll show you the supplemental table in a second. And it included, many of the causative genes that we all work with for cardiomyopathies and then neuromuscular disorders. Existing exome sequencing data. So some of the data here was actually reported in other papers as well. So they used a lot of data that they had on hand, was pretty nifty, and from the BioMe control cases. And then they also used raw genomic targeted panel sequencing data that they had. And then given the estimated incidence of acute myocarditis is 1 in about 10,000, and the estimated incidence of myocardial involvement with regard to viral infections being about 1 to 5%, they actually decided to exclude polymorphisms that had a minor allele frequency in the public database of whole genome sequencing from the population control. Furthermore, I'm going to go kind of more into the last two a tiny bit more in subsequent slides, but there were non-synonymous variants that were retained as a putatively deleterious variant. And they just kind of talk about what met the criteria for being a putatively deleterious variant. So as long as it didn't have a damaging functional effect determined as likely pathogenic or pathogenic. So obviously if it wasn't that, without a conflicting likely benign and benign entries among the badge laboratories in ClinVar. And then furthermore, this last point lists what they defined as the deleterious variants. So all of the in-frame and deletions and frame shifts and all of what met that criteria for deleterious variant and they use ClinVar as well. So here's the supplementary table with the 93 genes that were tested in both cohorts and controls. And then furthermore, like I mentioned before, they did do a viral analysis as previously described in other references. And then this other point talks about how the deleterious variant enrichment analysis and statistics were performed. So they discuss how they used a student's t-test, and they used that to compare the sequencing depth in the myocarditis cases versus controls. They used the Fisher exact tests that were two-tailed to compare the deleterious variant rates between these cases and the controls, and then so on and so forth. Now, they also, not only did they compare each cohort separately, but they decided that they would also pool them together. So they also did a pooled cohort analysis. And they talk about kind of a neat, what they termed as a resampling procedure. So they did, they pulled everything together and they had one set of data, and I'll show you those charts in a minute. But then they said to ensure the robust nature of their results that they performed this resampling procedure. And they generated a resampled odds ratio distribution. And they resampled subjects with replacement within each study, and they computed this overall odds ratio with a regression model, and they repeated the procedure 10,000 times. I'm sure they had a computer for this or a program, but they used this to kind of furthermore support their data. Now let's look at some tables. So for this table, this is just showing you our patient demographics. So for this case, it was a predominantly adult populated. So there were only 13 pediatric cases, and those were classified as people less than 20 years of age. So that was 11.1%. And then 104 of the cases were adult cases, so 88.9%. The majority of the subjects were European or Caucasian, self-reported ancestry, and they were mostly recruited from sites in Germany. So not that diverse of a study. Now, the genomic characteristics, this I kind of mentioned already, but it just talks about these 2 points are just going into more depth about the numbers that were that resulted from comparing the non-synonymous and synonymous variants, but in the AM cohorts, and the p-values were not significant. So this indicated that the AM cases and the control subjects, they were appropriately matched. And then when they also did across the case cohorts, it indicated that, these higher p-values indicated that these global characteristics were similar. So they weren't different. Now, as far as more results, so let's talk percentages. So the putatively damaging variants in genes that were related to these cardiomyocyte structures and functions, they were found to be enriched among individuals with acute myocarditis. So these were identified in 16.2% of all of the acute myocarditis cases. Okay, and you can see each separate cohort, 19, 11, and 31% in the AM1, 2, and 3 cases respectively. And when you compare that percentage to the control groups saw 7.2%. Now, overall, 3 of the 13 pediatric cases, so 23%, and then 16 of the 104 adults, 15%, those were identified as having more than one deleterious variant, but the p-value was very high, it was 0.44. Now, in monoallelic variants, those were present in about 17 of 19 cases that had deleterious variants, whereas 2 cases, they were both children, and they actually had multi-allelic variations. Now, the rates of these deleterious variants, those were similar between males and females, and there was really no difference in the median age of those with the deleterious variants and also the ones without. So this table is just showing you rates of the putatively damaging variants in the AM cohorts, but what I want to draw your attention to is that AM1 and AM3 have statistically significant p-values. So the deleterious variants, they were enriched in these cases, but only for AM1 and AM3. So just see my little red squares here. Now, this is the just a pictograph of and diagram of the pooled data. So using this logistic regression model in all of the 3 cohorts, there was a statistically significant enrichment of these deleterious variants in the pooled acute myocarditis cohort. And so our odds ratio was 2.5, and it was within the confidence interval. Now, a resampling procedure, like I mentioned, supported that analysis. And furthermore, the odds ratio was 2.48. So here's just a table that shows each cohort. So you have the top one, AM1, second, AM2, and AM3, and you can see pretty much all of the patients, their demographics, and their particular gene mutations. Now, among subjects with acute myocarditis, those deleterious variants were found in 13 of the 93 genes of the targeted panel. So that's kind of interesting. A single deleterious variant was noted in each of the 10 of these genes across the 3 cohorts. Now, 3 genes in particular were kind of the rock stars. They bore deleterious variants in multiple subjects, and these relevant variants were found in the DSP and the N of 3, DYSF, and TTN. And I have a nice table coming up on showing that. One TTN allele was actually shared among 2 subjects from the 3rd cohort, and those were subsequently found to be, they were siblings. Now, in figure 2, you can appreciate that you have all of the acute myocarditis genes and traditional phenotypes in this study. So on the x-axis, you can appreciate that these are the typical phenotypes: arrhythmogenic, dilated, hypertrophic, and neuromuscular disease. So this is what we would see. And then on the y-axis, you have the genes that kind of came up. Now, as the color darkens, the frequency increases. So you could see here, like TTN is dark black, and those were 8 cases. So the Mendelian phenotypes associated with the 13 acute myocarditis genes are listed as follows in the chart. So you can see with the arrhythmogenic group, you have 2, which is 15%. In the dilated group, you have 4, which is 30%. And then the neuromuscular disease is 61%. Now, although truncating variants in the TTN gene have been identified in 1 to 2.9% of the control populations in this particular study that they reference, the rate of this particular deleterious variant in this cohort was actually significantly higher. So they're comparing it to that. So it was actually, it was actually 6%. Now, when we look at the histologic type, and I think this is where everybody starts to salivate, right? Once we start talking about histology. So you have the genotype and phenotype associations here. Now, in this table, we have the histologic acute myocarditis types, and they divided into lymphocytic and non-lymphocytic. And the non-lymphocytic would include, of course, giant cell, eosinophilic, and granulomatous. Now, the detailed histologic data was actually available for 116 out of 117. Not too bad, right? Now, the deleterious variant status, so those with the variants, they were actually more frequent in the lymphocytic group. So 16 out of 87, 18% versus the non-lymphocytic group, which had 2 out of 29, which was 6.9%. But this association didn't achieve significance, p-value of 0.23. Notably, however, the AM2 group, which we had talked about before, the only cohort that didn't have a statistical enrichment of these deleterious variants, that one, greater 40% of the cases were considered non-lymphocytic. Okay, so that's an interesting point to take note of. This is a chart from the supplementary information. I couldn't help myself because I know this is what everybody wants to know, right? So for acute myocarditis, they used some criteria. They were looking for myocyte necrosis, and they were looking for, like I mentioned before, greater than 14 infiltrating leukocytes per millimeter squared. And that included the CD3 positive T lymphocytes. For chronic myocarditis, there couldn't be myocyte necrosis, and there would have to be also greater than 14 infiltrating leukocytes. For healed myocarditis, they didn't want to see necrosis, but they wanted to see fibrosis and partial presence of macrophages. For the dilated cardiomyopathies, they were looking for hypertrophic or atrophic myocytes, which is very nonspecific, loss of myofibrils and vacuolar degeneration, which is also nonspecific, severe focal and or diffuse interstitial fibrosis without inflammation. Now, for the results of the viral PCR, they were testing for adeno, HHV6 enterovirus, parvovirus, or the last column is if they didn't find a virus. So these viral etiologies were tested with PCR of myocardial tissue for 8 out of 19 of those subjects who actually were positive for a deleterious variant, and 34 of subjects who were negative. So whether they had a deleterious or did not have a deleterious variant, some still tested positive for viruses, and there was not a statistical predominance of any specific pathogen. So there are some outcomes data that's available for the AM1 and AM3 cohorts, but not AM2. I'm going to show you the next slide. There were 5 out of 11 subjects who had the deleterious variant from these 2 cohorts who actually either died or they required transplant or LVAD, versus 14 of the 35 subjects who were actually negative for the deleterious variant. So this is also in the supplementary information, and this is showing you all your patient demographics, and they actually give you each patient's baseline left ventricular ejection fraction at baseline, and then you have the follow-up 1 to 2 years later, and then they could determine whether or not this ejection fraction improved. So in the third cohort, where the ejection fractions during acute illness or follow-up were available, 2 out of the 6 subjects who had deleterious variants, they did experience what they called persistence of cardiomyopathy versus normalization of function. And then 11 of 14 who didn't have these variants, they actually had what they called chronic cardiomyopathy versus recovery. Among all the subjects where we had ejection fraction data, these deleterious variants were found in 12 of 55, so only 22%, with an ejection fraction less than 40. Okay, so it kind of correlates with a worse ejection fraction versus 2 of 28, which is 7%, and those who had a higher ejection fraction. That kind of makes sense. Now, of the 2 children that had multi-allelic deleterious variants, the histology revealed lymphocytic acute myocarditis, and myocardial viral RNA and DNA was actually not detected, and the outcomes were transplant in the first patient and ventricular assist device in the second. So let's get into the discussion. So based on all this data, some of the introduction we talked about, it seems as though that the risk for development of acute myocarditis is likely multifactorial, caused by both some sort of environmental exposure and an underlying genetic vulnerability of the substrate, whether it be cardiomyopathy genes like we're talking about, but there are other modifiers that also could include something such as the gut microbiome, vitamins, trace elements, and these all have been well described in animal models. The particular interplay of the virus and a host genetics is actually highlighted by previous rodent studies, which demonstrate that these genetic dystrophin deficiencies increase in enterovirus induced cardiomyopathy, and that Coxsackie virus B3 targets the cardiomyocyte dystrophin glycoprotein complex to impair the sarcolemmal integrity. So there's a lot of mechanisms here, and I'm sure you guys have heard of it, the CAR receptor, that are involved as well. And that kind of goes on the whole side of like the immune side of things, which we'll kind of touch on a little bit in the second article. Now, shifts towards these genomic paradigms have recently occurred for several other myocardial diseases that were previously attributed to environmental toxic or immunologic factors. I won't read you all of this, but they give two examples in the paper. The first example that they talk about is pregnancy-associated peripartum cardiomyopathy. And so they cite some studies that found that a small percentage of women with this peripartum cardiomyopathy did harbor some rare truncating variants in 8 different cardiomyopathy genes. And this was seen similar to rates in those patients with dilated cardiomyopathies. So one of the most frequently implicated genes was the TTN gene. So heart failure guidelines have actually recently been updated to acknowledge that these results exist and that genetic testing is recommended in this situation. The second example they gave was chemotherapy-induced cardiomyopathy. And so this is a complication in 9% of patients who are treated with anthracyclines, for instance. And most of the time, clinical factors don't always account for the susceptibility. I mean, you can talk about concentrations and everything, but that's not always the case. The more, the worse. So several recent case studies and series, they have illuminated the role of genetics in this position, and potentially they've identified these pathogenic variants in the TTN gene and MYH7 among affected individuals. So those data suggest that a genetic substrate plays a critical role. This 2-hit hypothesis may extend to acute myocarditis. So this infectious exposure would be considered like a second hit, because your baseline or your first hit is going to be what you are born with, what's going on in the heart muscle. And the author's previous work and that of others does support the 2-hit theory. The current investigation extends this as one of the largest genomic investigations of patients with acute myocarditis to date, studying mainly adults. Now there's more, lots more studies and lots more kind of examples of these genes coming onto the scene. The authors did find significant enrichment, as we've talked about, in these damaging variants with when compared to these controls. But the rates of the variants, they weren't actually statistically different between pediatric and adult cases, even though we had, very small amount of pediatric cases. Most of the relevant variation was heterozygous, but there were 2 pediatric cases which harbored multiple deleterious variants. And then although the author's prior exome sequencing study of kids with the acute myocarditis had identified some homozygosity and compound heterozygosity, double heterozygosity may also confer a higher risk in acute myocarditis. So this is something that's being described. 3 genes, DSP, DYSF, and TTN, these are implicated in multiple subjects. And these putatively damaging variants in DSP, along with PKP2, many may know PKP2 as an offender in arrhythmogenic cardiomyopathy. These were also identified among subjects in the author's prior pediatric cohort. DSP has been implicated in several other recent case reports. The genes observed in this study are traditionally associated with the phenotypes that we discussed, and with this final common pathway framework, these findings do suggest that impairments in the structural integrity of these myocytes render them susceptible, and I do agree. And as far as these subgroups, even though they were small, the authors, they didn't observe, any statistically significant associations between that actual deleterious status and either the presence or absence of the genome. But although there was a trend towards higher rates of these deleterious variants in that particular group that was just lymphocytic. Interestingly, in one of the cohorts where the deleterious variants weren't statistically enriched, which we had that chart, it was the non-lymphocytic histology. In 5 of those, the deleterious cases, they were in subjects that had that specific histology. Now, the trend of the deleterious variants was seen less frequently in the giant cell granulomas and eosinophilic cases, and this suggests that maybe there are mechanisms for susceptibility to such forms that may fall outside this final common pathway. I don't really know what to say about that. Overall, though, this study was not powered to assess clinical outcome, but they did have some data, that did provide some clinical outcome. But the findings, they say that the findings suggest that the presence of these variants may not accelerate or worsen the course. Future studies need to be done with larger numbers of patients with acute myocarditis with more uniform clinical data. And we need to clarify whether is it that this status of having a deleterious variant, is it really just a hit in this 2-hit mechanism, or is it something that's altering the outcome? So is it causing maybe more a brisk inflammatory response, or thinking they've had an inflammatory response before? It's just that threshold that's getting crossed. Recognizing these underpinnings, it may be useful to really kind of dive into these mechanistic insights, but that would be a whole nother lecture, actually. The lack of the association with any specific clinical features suggests that only genetic testing can uncover these cases. I can understand why they would say that. Now, for both the autosomal dominant and recessive forms of genetic acute cardiomyopathy, the family screening may be warranted. So that's something I think, especially me coming from a medical examiner's kind of office, a lot of times, we get cases of, okay, we don't know what's wrong with this heart. We run genetic testing, or we order molecular testing, and a couple days we get our histology back. And we see myocarditis. So, I mean, I've canceled molecular testing before because it's like, well, we have a cause of death now. We know what's going on, but maybe we're shifting. Maybe we need to actually do the testing as well if you can. Just to see if there is a genetic component that may be significant for the family to be aware about. Now, as with peripartum cardiomyopathy, this clinical genetic testing for acute myocarditis may be one day recommended within society guidelines. And based on these findings, a panel that comprising both these cardiomyopathy and neuromuscular disease may be of the highest yield. And I know that, it just depends on what targeted panel you're getting, but not all of them are going to have the neuromuscular disease genes. So some of the limitations in this study we've already mentioned. There are differences in the extent of data available within the 3 cohorts. The detailed histologic data was only available for 2 of the cohorts, and all 3 cohorts comprised individuals predominantly from European ancestry. So these genomic findings in this study, they may not be applicable to a more diverse population. And there are studies, I have read that there are some studies ongoing, with different ethnicities. Now, some last points I'm going to just mention, and then I don't know if we want to have people chime in or if I just keep going, but I think, there is utility for more comprehensive testing, like I mentioned before, not just, canceling your molecular test if you find myocarditis. Also, just a note, and I think Dr. Boyes can appreciate this point with viral testing. Viral testing is something that not everybody does. It's not because we don't want to, it's because it's not really commonplace in a lot of institutions. And we all know that in some of the guidelines, it is recommended that if you have a myocarditis case, you do a endomyocardial biopsy, you send a piece of fresh tissue for viral testing. But it's not done everywhere. It's not as easy to get the test validated in some institutions. So I do think it's a challenge, but I do think it's something that I think universally, a lot of institutions would appreciate working on, maybe collaborating and getting that kind of revved up, because I think it would be really useful. Of course, I have to highlight the importance of autopsy studies. I know everybody here, most people love autopsies. It's such a rich environment to have tissue and actually be able to use it for a really, really good purpose. So you have plenty of tissue at an autopsy. Now, the issue would be, are you going to be dealing with fresh or formalin fixed tissue? And both, you can use this for, genetic testing or even the viral testing. So I do think that, the material is there, so to speak. And then lastly, I would love to hear from the more experienced cardiopathologists out there. What are your thoughts? Have you guys discovered similar findings in your daily casework? And kind of where are you in this kind of, in this new data and how it's being presented? Like, are you guys on board with this idea? And what did you think about this paper? So Dr. Wang, would you like us to kind of take a little moment to talk about this one? Or
I kind of suggest we keep going because
Okay.
we don't have much
Okay.
time. And yeah,
Okay.
and then we will keep all the discussion later on.
Okay, sounds good. All right. Okay. All right, take a deep breath. Okay, next one. So this one is a paper that discusses pathogenic variants in some of the cardiomyopathy genes that underlie pediatric myocarditis. So somewhat a similar situation, except in kids. And then furthermore, this added little nugget, a further impact of heterozygous immune disorder gene variants. So in the pediatric setting, we know that myocarditis is mostly common in children and of under 2 years of age. And a lot of times these kids will develop a dilated cardiomyopathy, and more than 50% that present with a very severely impaired ejection fraction or left ventricular cardiac function. And the clinical courses of these children can be very severe. We know that there's increased mortality, there's a frequent need for mechanical circulatory support, and then sometimes heart transplantation. Now, pediatric dilated cardiomyopathy, if you just look at it, as its own thing, it's a primary form of cardiomyopathy in children, and it does have a peak incidence in the first years of life as well. So taking that in mind, as there's a little difference between these two entities, if we're looking at both of them head to head, differentiation on the basis of clinical data alone is very difficult. This usually requires further diagnostic validation with an endomyocardial biopsy. Now, more frequently, pediatric myocarditis cases diagnosed with a positive biopsy, they're affected by healing or chronic myocarditis and substantial ventricular modeling. And usually these kids do need mechanical support, circulatory support, and in about 20% of cases, and then transplant in about 10%. So mechanistically, we touched on the whole idea of the different pathways of the immune system, but carditis does involve the innate and adaptive immune system, and these components become activated after viral entry. There are common receptors. So for instance, the Coxsackie adenoviral receptor, the CAR receptor, there's the decay accelerating factor receptor, the DAF, and then activation of your innate immune response. And so toll-like receptors, so TLR3 and 4. Now, consequently, natural killer cells, macrophages, then they come and they get recruited to the myocardium. They release the interferons, and it initiates this lymphocytic proliferation and activation. Subsequently, these T lymphocytes, they do target viral proteins, but they also expose a mimicry mechanism towards myocardial proteins. One of these such proteins is the myosin heavy chain. This suggests that myocarditis is an intertwined pathology of immune defense against external pathogens and autoimmune response to myocardial antigens. The frequent association of dilated cardiomyopathy with inflammation, this is implicating a critical predisposition of the myocardium. So recently, these authors showed that pediatric myocarditis with a dilated phenotype is due to pathogenic or likely pathogenic variants in certain disease genes. In that particular study, the clinical group, it's interesting, they were able to clinically differentiate the myocarditis cases with a dilated picture and the myocarditis cases that did not have a dilated picture. And the ones that were dilated had a very worse outcome. And most of the kids that didn't have the dilated phenotype, they recovered. However, the patients in the dilated group, they often required mechanical circulatory support or transplant, and they more frequently carried likely pathogenic variants. Now, this suggests that a proportion of pediatric patients with myocarditis that they have some sort of underlying primary genetically determined dilated cardiomyopathy. So I'll skip through this, but this slide kind of highlights there's a whole list of more studies where they're showing the same thing. And we kind of touched on some very similar stuff in the other paper. Now, altogether, this suggests that there are some genetic defects of immune system components that are heterogeneous. And the impact of these hetero, hemi, and homozygous genetic variants in a broader spectrum of known immune diseases, it hasn't been systematically explored in mono or oligogenic traits so far. So the authors have a hypothesis. They want to test this hypothesis, the hypothesis that patients that have a severe dilated cardiomyopathy with biopsy-proven myocarditis, that they are carrying the genetic variants in these cardiomyopathy genes and immune disease genes. And they also want to utilize a family-based genetic approach. So they actually, as I will mention, they have an index case, and then they further test the family. So for the method, so the study population, clinical data from patients were from below 18 years of age that had suspected myocarditis, and they were extracted from the medical records. Starting in 2013, 6 of those patients were also enrolled in another kind of a clinical trial registry called the MYKKE. And this study also used a family approach using genetic analysis of both index and respective parents. Now, the inclusion criteria were the following. So they had to have biopsy-proven myocarditis, and they had to present with a dilated phenotype. And that's what we mentioned before. So the exclusion, so they excluded people in the study, so patients that had any structural congenital defect, any syndromic disorders, situs inversus, anything metabolic, mitochondrial, or neuromuscular. And clinical and diagnostic assessments, they included all the laboratory parameters, the cardiac imaging, and the follow-up was also performed for the occurrence of any major cardiac event, whether it be an arrhythmia, did they need a transplant, did they have to get an LVAD, or did they die? Now, the study was conducted according to the guidelines of the Declaration of Helsinki, and then all the appropriate consents were performed. So for their statistical data, they used categorical variables summarized by frequencies and percentages. They used Pearson's chi-squared test to compare the different dichotomous variables. Now, when looking at the biopsies, these were taken according to clinical routine. So usually we get them from the right side, but they got some biopsies from the left. They got it also from the right, or they sometimes would get it from both ventricles. Or they called it a transmural specimen during the VAD, or ventricular assist device implantation. So the little plugs when they put the device in. And all of these biopsies were characterized by histopathology and immunohistology as previously described. Then they also did qPCR for viral RNA and DNA. And the histological analysis was followed by the Dallas criteria, and this was the gold standard. And this was completed by different IHC staining. And the diagnosis of myocarditis was confirmed according to the established criteria, categorized in accordance with the WHO definition. Now, for the next-gen sequencing, they isolated DNA from the peripheral blood. They did use whole exome sequencing, and they go into more detail on the enrichment probes, but they did create a library. And they did 89 disease genes. These are all the variants. Now, when you look at the ones that are in a regular format, those are the genes that were considered disease causing only if they were autosomal dominant. The ones that are in bold are considered disease causing only with an autosomal recessive inheritance. And the same for this. This is actually the gene, the laundry list of immune genes that they tested. So for the genetic analysis and variant classification, all of these filtered genetic variants, they were classified as either pathogenic, likely pathogenic, or VUS. according to common guidelines that we all know about. Now, the genetic variants that they detected in the index patients, they were traced in first-degree family members, and so that they could determine, like, is this a de novo mutation or segregation. They also did this combined annotation dependent depletion score, the CADD score, which was calculated. And this is actually a more of a quantitative prioritization approach for any genetic variants, implementing multiple computational predictions. You can read more on that if you want. Now, the score actually ranges from 1 to 99. Now, the higher values indicate a more deleterious impact of a genetic alteration. So from 1 to 99, 9 being the worst. The threshold greater than 10 to 20 indicates deleterious substitutions. So here's a little pictogram. It's showing you just the flow. We have genetic evaluation after a whole exome sequencing, and it includes patients with the biopsy-proven myocarditis and the phenotype of dilated cardiomyopathy. Now, the genes, both cardiomyopathy and immune genes, these are filtered with minor allele frequencies, whether they be, pathogenic, likely pathogenic variants. And then they also furthermore looked at the heterozygous versus homozygous and hemizygous variants. And these all include, and then they go into the disease that included missense protein length changing and splice variants. And this is all in ClinVar. So it's kind of the same thing that we mentioned before. Now, for the results. So for clinical characterization, there were 12 families, and that included the index patient and both parents. The index patients had a median age of 1.6. There were 5 males, 7 females, and all of the children presented with this dilated phenotype. The mean left ventricular ejection fraction was 23. All had heart failure symptoms, all received medication. They all needed inotropic support. For immunological events, they described that a viral respiratory infection was seen within 6 weeks before admission in 7 patients. One of the patients in the study also received vaccination against the tetanus, diphtheria, whooping cough, influenza B, polio, Hep B, about a week before they were admitted. And then there was another patient who was of small gestational age, and this was family 5. And this patient was later in life diagnosed with morbus Crohn. And then severe underlying immune disorders were excluded in all patients and parents during this in-depth assessment. And then in all patients, like we've already mentioned, they had biopsy-proven myocarditis. Acute myocarditis was seen in 1 case, chronic or healing myocarditis in 8, and then unspecific macrophage-dominated inflammation in 3. Myocardial virus was detected in 6 out of 12, and 2 were in parvovirus. HHV6 had 4, and most of these patients had low viral loads. 11 patients did undergo VAD placement, and then 9 patients actually had a transplant, and none of them died. One of the patients even was weaned off of his VAD. And they also mentioned that this particular patient had moderate viral parvovirus levels. So this is just a table detailing all the patient characteristics, the New York classification, all the lab findings, echo, and you can see that none of the patients died. So they talk about all the complications in all of the devices. So in these analyses of the 12 families, there was a likely pathogenic variant in cardiomyopathy genes in 8 out of 12 index patients and related to dilated cardiomyopathy. And assessing the genetic variants in the 89 disease genes, it yielded 1 likely pathogenic variant in 8 patients and at least 1 VUS in 3 patients. And there was no, in 1 patient, they actually had no genetic cardiomyopathy variants. And the index patient of family 10, they kind of go into more of the specifics. They mentioned how it was a compound heterozygous genotype of 2 VUS missense variants of the FKTN gene. And it was actually inherited in trans from the mother and the father, which explains this phenotype of a skeletal myopathy. They also mentioned that from the 8 likely pathogenic variants, 6 were actually missense and 2 emerged as a stop gain. And then lastly, I'll just touch on the last point that the high yield of these cardiomyopathy likely pathogenic variants is reflective of the severe clinical stage of the analyzed pediatric cohort. So for the genetic analysis, more specifically, let's talk about the immune genes. So we have 631 immune genes, and it yielded at least a VUS in 9 of the patients and no genetic immune variant in 3. 15 variants were detected overall, 15 out of that large laundry list. All 3 of the likely pathogenic variants were actually annotated in ClinVar as a likely pathogenic, and they were in the heterozygous state. And those had CADD values of greater than 30, and they represented stop gain variants. And of interest, some of these genes were actually noted to be in these primaries, like a ciliary type gene group, and they were indirectly associated with ciliary transport. So these variants were associated with recessive inheritance of Fanconi anemia, complementation group C, primary ciliary dyskinesis, and primary ciliary dyskinesia. So considering that there's the molecular function of immune disease genes, they really did note that there was an accumulation of these genes, whether they were directly or indirectly associated with ciliary transport. So we're kind of getting low on time. All of this stuff is just more specifics on these family pedigrees and the index patient, and how where the genes were inherited from the mom or the father. I don't know, Dr. Wang, if we should just go ahead and have some discussion.
Yeah, I think.
Yeah. Okay.
Yeah, some discussion is actually these are 2 very interesting paper. So some discussion, I think is really kind of we can hear what people feel.
Right.
So any questions? Any discussing point from our audience? So while people are waiting for, I look at this paper. This viral infection rate is pretty high, right, in both study?
Yes.
Compared to many institutions.
Yeah, and I think even in the second study, more so. I was actually pretty surprised.
I had a question. It seemed like, at least in the first paper, that the mutations were both in structural and ion channel genes like SCN5A, and it seems odd that there would be such a heterogeneous amount of mutations that would lead to a common vulnerability to myocarditis. I don't know if anyone has a comment on that, but in other words, if it was all a structural gene like Titin, as many of them are, or myosin heavy chain or myosin binding protein, okay, that's makes more sense. But to have to have other genes or other proteins as well, such as ion channel genes, it seems like this needs more work to refine it.
Mm-hmm.
Yeah. So I don't. Melanie, you have a questions?
No, I was actually just going to comment on Dr. Cole's analysis too. I mean, I do feel as though, there's Sarah, first of all, great presentation, a lot of material and a lot of really great information, and you did a very nice job kind of showing it all to us. And I agree that it is interesting that there's clearly different genetic underpinnings with a common pathway. And I think it really underscores the fact that we do need more work in this particular area. I do think that some of the studies that we've laid out nicely show that there may very well be a genetic component, but what those exactly are in their effects, I'm not entirely sure that we're clear on that yet.
I agree. It was an excellent presentation. And just one quick comment as a forensic and cardiovascular pathologist in a medium-sized practice for forensic cases. Myocarditis is vanishingly rare. We get it one case every 2 or 3 years, whether it's children, more adults, I guess I'd say, than children, but still occasional kids. But to get enough cases, as I guess was proven by these large multi-center studies where the pediatric one had 12 cases. I think it's going to take a lot of work to power the statistical analysis to where it's more convincing. Although I agree, this is very cool. I didn't know, I had never thought of this. And I think I will also ask for genetic studies when I get my next myocarditis case.
Here we go.
Yeah, I mean, I think there's so much to kind of unwrap here. I mean, not only are we talking about, the molecular components of the actual muscle itself, but, as mentioned, the environmental factors, nutritional status, ethnicity, more populated, diverse areas probably see, higher rates of myocarditis. But there's a lot to unpack here. I just think it's quite interesting. And then, even with regard to, let's talk about, like arrhythmogenic cardiomyopathy, many times, a lot of times we do see an inflammatory infiltrate. So some of these folks are biopsying maybe what some of us have called the hot phase, but, the whole concept of viral testing, whether or not it's going to be positive, if it really is a virus or not, or are they bystanders, it's still very blurry. I think we're still figuring things out. But I do think, that I agree with you, Dr. Coley. Like, I'm super excited. The next time I get a myocarditis case, I want to definitely do molecular testing. So.
And you can combine, so you can get big number.
Exactly. I just got to see if we have the funding.
See,
yeah.
So, Sarah, so we're talking about this screening process, and you'll find the 3 genes in the first paper, right? One
Yes.
is DSP, another TM.
Yes.
Anybody look at longitudinally, say, the people have this one. How much of them will have myocarditis?
That's a good point. No, that's a really good point. And it's like, you want to know, are they in folks that are alive? Or are they in folks that are dead? And if so, if they're in folks that are dead, how was the heart sampled? Did they just put one cassette in, or did they put a few? Because as we all know, myocarditis can be very patchy. So that's a very good point.
Any more questions?
Well, great topic and great discussion.
I think these 2 papers are really interesting. We need, there's too many information. There's so many information.
I know.
Digest it a much longer time, and then we come back to some really kind of practical approach.
I agree with you. And when I was putting this together, I was like thinking maybe I should just do one paper, because even just one paper touches on so many different ideas. There's a really wonderful article in Nature that I don't know if any of you have read it. It is outlining myocarditis kind of from a global standpoint and all of the future directions. But that's another paper that I came across in this search, and it kind of goes into a lot of these little facets. So this predisposition part of it, the viral component to it, and all of the other factors like the mechanisms. So all of the molecular biology and the research that needs to be done. So if any of you are interested in that paper, I can send you a copy. I would be glad to share that.
Yeah, can you send me those papers? I'm really interested in that.
Yeah, I'm interested as well. I don't know if you need my email.
I think I could find it. I can find that. Yeah.
Yeah, thank you so much, Sarah.
Yes.
I really appreciate.
Awesome.
So thank everybody for joining us today, and we'll continue next month.
And thanks, Peter. Thanks for having this.
Thank you.
Thanks, Peter. Thanks, Sarah.
Thank you.
Bye. Good to see you.
Okay. Adios.
Bye, guys.